So that was the mitochondria. So in mitochondria we have Kreb cycle, oxidative phosphorylation, that's on the inner mitochondrial membrane. Then we have some part, for example, of the synthesis of steroid hormones, some part of synthesis of heme, and so on. Yes, so many pathways. Here you have the list. I think that all groups has some kind of this list. Yes? So what other compartment you know? Golgi apparatus, yes. What's localized? Which pathways are located there?
I started saying like the rough endoplasmic reticulum, and I decided to include the Golgi as well.
So we have the nucleus, rough endoplasmatic reticulum, Golgi apparatus. In nucleus we have the replication, transcription. On ribosomes and the rough endoplasmatic reticulum we have translation. Then in Golgi apparatus we have the post-translational modifications of proteins and so on. Yes? These three organelles are more molecular biology than pure chemistry. Yes? So what, for example, in cytoplasm, what do we have there?
Fatty acid synthesis.
So synthesis of fatty acids, what else? Glycolysis, what else?
Gluconeogenesis.
Yes, gluconeogenesis.
Part of synthesis of urea.
Yes, part of synthesis of urea. Then also part of synthesis of heme and so on. Then there are purines, pyrimidines, also part of them. Metabolism of glycogen. Yes? So in the presentation you can read the other compartments, lysosome, peroxisomes, yes? So read it and remind what you have learned in the winter semester, yes? In general, the compartmentalization is important for we have many compartments in the cell, and in every compartment there are specific reactions. For example, in metabolism of lipids, in cytoplasm we have synthesis of fatty acids. In mitochondria we have degradation of fatty acids. Yes? So it means that in one compartment we have synthesis, in other we have degradation, and therefore the fatty acids that are synthesized, they cannot be directly degraded. Yes? This is the important thing. If we have some subsequent processes, for example, Kreb cycle and respiratory chain, they are in one compartment. In mitochondria, in the matrix, and on the inner mitochondrial membrane. And under the term compartmentalization, we should also think about the transporters between two compartments. Yes? For the cell is one big compartment that has the membrane. So if you want to put something into the cell, you have to have transporter. Yes? For example, sodium glucose transporter or GLUT4 transporter. And the same transporters has to be on the borders between two compartments. For example, do you know some transporter that is important in the inner mitochondrial membrane? What? Carnitine transporter, yes. So you know that if you want to degrade fatty acid, the fatty acid has to enter the cell, and then from the cytoplasm to the matrix is transported by the carnitine transporter. And not only enzymes, but also these transporters can be regulated. And you know that this carnitine transporter consists of 3 enzymes, and one of them is regulated. Or the GLUT4 transporter is not normally in the membrane, but only if you have insulin in your blood. Yes? Do you understand that also the transporters can be regulated? Not only enzymes, but also the channels or transporters can be regulated. And therefore, the compartmentalization plays so important role in the regulation of metabolic processes. Yes? So not only the border, the membrane, but also the transporters. Yes? You know that, for example, the derivatives of coenzyme A cannot go through the membrane. So we have to have the transporter. The same is with other big compounds. They need to have the transporter. Also, for example, the reduced cofactors, they need to have the transport mechanism from the cytoplasm into the mitochondria. So that's the compartmentalization. The second topic is how to change the absolute concentration of the enzyme. It means the synthesis of new enzyme molecule or the degradation of the enzyme molecule. Yes? So how to synthesize a new molecule? How is enzyme produced? Yes, so we have some gene, we have transcription, translation. Do you know how this can be regulated, the transcription and translation, how it's regulated in our body?
For example, signaling by hormones or something?
Yes, and the step between the hormone and the transcription is some factor that is called transcriptional factor. Yes? So we have transcription factors, and we are speaking about induction or repression of the transcription. Yes? In molecular biology, you have learned many transcriptional factors. For biochemistry, it's important that these transcription factors have some sites where you can bound some ligand. Then we have the ligand-dependent transcriptional factor. And you have to know that these ligands can be not only signaling molecules, but also some metabolites. And then if you have some metabolite that is inductor, or that if you have some metabolite that bind to the transcriptional factor that is inducer, you can say that, for example, xenobiotics induce the production of monooxygenase system. Do you understand? What does it mean? You have some metabolite, for example, some xenobiotic, yes, some anthracene. And this molecule is able to bound to the transcriptional factor. And by this way, it can increase the expression of some enzyme. Or the ligand can be signaling molecule. Do you remember we have one group of hormones that can increase the synthesis of catecholamines? Do you remember how they are called? It was in the previous lecture. Not this. In the adrenal cortex, we produce glucocorticoids. And these glucocorticoids go into the medulla, adrenal medulla, where you synthesize catecholamines. And these glucocorticoids are very potent inducer of catecholamine synthesis. The same is the xenobiotic. If you have a lot of xenobiotics in the cell, you increase the production of the monooxygenase system, the enzymes that degrade xenobiotics. Or you can do the repression. Do you know some metabolite that by the feedback inhibition repress the production of some enzyme? It was also the last lecture.
Citrate synthase?
And citrate synthase is by what?
By what enzyme or?
What metabolite suppress the production of citrate synthase? I do not know whether, I don't think so that the isocitrate, that citrate synthase is regulated by this.
But it's some feedback there, isn't it?
That's the feedback, yes, but not the feedback by the reduction of the expression. For you have the short feedback is when the metabolite decreases the activity of the enzyme. But now we have the feedback when the metabolite goes to the nucleus and there decrease the expression. That was cholesterol. If you remember, if you have a lot of cholesterol and a lot of metabolites, they can go into the nucleus and there they can decrease the production of hydroxymethylglutarate coenzyme A reductase. So cholesterol is the typical molecule, typical metabolite that repress the expression of enzyme. The same do purines and pyrimidines. If you have, for example, AMP, it's able to suppress the production of new enzyme. This regulation, it means the production of new hormone, new enzyme, it takes hours or days, yes? So it's a slow mechanism. If you want to produce a new molecule, new peptide, new enzyme, it durates long time. It means hours to days, yes? The modulation of the activity, that's quite fast. It takes seconds or minutes, yes? But this regulation, due to the change of the gene expression, it takes long time. The second subtopic is how to destroy the enzyme in the cell. How we can destroy the enzyme. I don't understand. Ubiquinone, yes. Yes, so that's the proteolysis. We have enzyme, and when we want to destroy it, we need to do a proteolysis. And we have a special molecule that is called proteasome, where we can degrade all proteins. And the mark for the proteasome is called ubiquinone. So if you want to degrade some enzyme, you put there this mark, and the enzyme is degraded. And different enzymes have different affinity to this mark, to the ubiquinone. Yes? So enzymes are degraded in proteasome by proteolysis, and the mark is ubiquinone. And different enzymes have different affinity to this mark. The third topic is the modulation of the activity of existing enzyme. It means we have some enzyme and we modulate the molecule. And there we have two possibilities. The first is the covalent modification of enzyme molecule, and the second are interventions that modulate the enzyme kinetic. So the covalent modifications. What do you know about the covalent modifications of enzyme molecule that activate the enzyme? Phosphorylation or dephosphorylation, and the second mechanism is
sulfide bonds?
What?
In case of insulin, cleavage. And
yes, so limited proteolysis, you mean. The second mechanism is the limited proteolysis. It means you have the inactive molecule and you chop, for example, 4 amino acids and you produce active molecule. So the inactive molecule was called proenzyme or zymogen. This is the first possibility. This is irreversible. So you have big molecule that is called proenzyme. It's inactive, and you can do a limited proteolysis. You take few amino acids away, and you gain active enzyme. This is irreversible, yes? You cannot inactivate this enzyme. And the examples are, for example, digestive enzymes or the enzymes of blood clotting. Yes? We have pepsinogen in stomach, and the active enzyme is called pepsin. The same, we have trypsinogen, and the active enzyme is called trypsin. And the whole blood clotting is only about this. We have inactive precursors, about 10 inactive precursors in our blood. And then there is the first signal that activates the first enzyme, and then in the cascade, they activate the second, third, fourth, and so on. And all are proteases. The second mechanism of covalent modification is the phosphorylation and dephosphorylation. That's a very fast mechanism. It durates milliseconds or seconds to change the activity of the enzyme. And how it works. We have enzyme and we need some other enzyme that is called kinase. Protein kinase, you know that we have many kinases. But all kinases do only one thing. What's the reaction? How you can write the reaction?
Phosphorylation.
They take phosphate, they take protein plus ATP. They produce ADP and phosphorylated protein. Yes, so that's simply a reaction. You have protein kinase, and all take ATP and put phosphate on the peptide molecule. Yes? The opposite process is dephosphorylation catalyzed by phosphatases, protein phosphatases. Yes? So they take protein with phosphate, they do hydrolysis, and you have protein and inorganic phosphate. You know that some enzymes are activated when they are phosphorylated, some they are inactive when they are phosphorylated, and this you have to learn, yes? That, for example, glycogen synthase is active, phosphorylated or dephosphorylated?
Dephosphorylate.
Dephosphorylated. And glycogen phosphorylase, opposite. Yes? So this you have to remember. But if you know that insulin induces dephosphorylation and glucagon induce phosphorylation, it's much easier to remember it, yes? Insulin, anabolic hormone, dephosphorylation, glucagon, catabolic hormone, phosphorylation, yes? The precise mechanism between the glucagon and its receptor and the glycogen synthase or phosphorylase, you know. Yes, many kinases and so on, so on. So that I think was the, not. That was, this is one cascade. And the last thing before the break, we should speak about the interventions that affect the enzyme kinetics. About that you had one lecture. So I hope that you know it. The first, there are two groups of these interventions. The first is that you change the concentration of substrate or the concentration of product, or you change the temperature, pH, and so on. Yes? The second mechanism is that you put there the compound that is able directly to change the activity of the enzyme. Yes? Here we have the activator and inhibitors. When we were speaking about the synthesis of new enzyme, there it was inducer and repressor. Yes? So these terms are different. Activators are and inhibitors. They change the activity of the enzyme, and inducer and repressor, they change the transcription and translation. So you know that the enzyme needs to have the substrate. Then there is some maximal point when all enzyme molecules are occupied, yes? So now you can add 10 moles of substrate, but you cannot accelerate the reaction, yes? That you understand, that there is some maximal velocity when all enzyme molecules are occupied, yes? If you want to accelerate the reaction, the second possibility is to remove the products. Why removal of products is able to accelerate the reaction?
Because the concentration, like there's a greater concentration gradient of the finished product from the part associated from the enzyme complex.
Mm-hmm. So then it can be faster.
Yeah.
Yes. So if you are able to remove the products fast, then the enzyme tries to keep the equilibrium, to return the reaction to equilibrium, and therefore the reaction is faster. How to remove the product? The first possibility is to have a long metabolic pathway when the product of one reaction is substrate for the second reaction. Therefore, we have metabolic pathways. Yes? The first reaction, the product is glucose, and product is pyruvate. And we have many steps, but all are connected to one line. And therefore, the glycolysis is fast. The second possibility, how to remove the product, is to take it away, for example, to other compartment, or to pump it away from the cell. There's a second possibility, how to remove the product. Each enzyme has the temperature optimum, pH optimum. That's important to know. The majority of enzymes, they like pH between 7 to 7.4. Yes? pH inside the cell is 7. In our blood is 7.4. Yes? But there are some exceptions. For example, digestive enzymes. Pepsin likes to have pH 1.5 and trypsin about 8.5. Yes, that's the best pH for them. But the majority of enzymes between 7 and 7.4. If you have different pH, what would happen to the enzyme molecule?
It's inactive.
And why it inactivates?
Because it doesn't bind to the substrate.
And why it doesn't bind to the substrate?
Because it's not ready.
Denaturation, that's the last step. You absolutely destroy the structure, but before the denaturation,
yeah, the structure is modified.
yes, to keep the enzyme structure, you have to have the proper functional groups on proper places. Yes? Some functional groups are dissociated, some not. And if you change the pH, you change the dissociation. And therefore, the enzyme change the structure, also change the ability to bind the substrate. Yes? And therefore, the enzyme needs to have the pH, some optimal value of pH. If you change this, you change the dissociation of functional groups, and you change the function. And the same is with the temperature. Yes? When you change the temperature, the enzyme changes the shape. So that was temperature and pH. And then we have the activity modulators. You had one special lecture about the inhibitors, so you know what is it, competitive, non-competitive inhibition. Only I would like to remind here the term feedback regulation, cross-regulation, and feed-forward regulation. So what is it? Feedback regulation. Yes, so the product inhibits the first or second reaction, or the regulatory reaction. An example? Yes, citrate synthase and what?
It's inhibited by citrate, I guess.
It's inhibited by citrate. But that's only that one reaction inhibited by the product. So something longer. If you have one metabolic pathway, one long metabolic pathway, you have substrate, product, and the product inhibits the first or second reaction.
I have a question. But does feedback regulation only have to be negative, or can it also be?
It can be also positive.
Because then I was thinking about in prolactin, the more.
You mean more?
In breast, like when mothers
Yes.
breastfeed, the more they breastfeed,
Yes.
the more prolactin will.
Yes, that's true. That's the milk ejecting reflex. Yes, then there it's positive.
Yeah.
The same positive, but that's not on the cellular level you have during the birth. So the first contraction increased the levels of the oxytocin receptors. Yes, but now that's true. We have the feedback can be also activation and inhibition. Yes, that was the important note. But tell me an example. When you have feedback, inhibition, the product inhibits the pathway.
Cholesterol.
So cholesterol directly is able to inhibit the hydroxymethylglycerol coenzyme A reductase. There's one mechanism. But also cholesterol is a repressor of the transcription. These are two independent mechanisms. Add the other example. Yes, how it works.
If you have a lot of ATP, you inhibit it.
So if you have glycolysis, the regulatory enzyme is phosphofructokinase, and this enzyme is inhibited by ATP. Why? For if you have a lot of energy in the cell, you don't want to produce new energy. And after glycolysis, you have Kreb cycle and so on. But that was true, but the end product of glycolysis, the name is pyruvate or lactate. Yes? And these two compounds can also inhibit the reactions. How they do it?
Lactate, for example, it's acid, so by its pH, then it inhibits the enzyme.
Yes. So the glycolysis, at the beginning you have neutral molecule. At the end you have acid. If you have very, very low pH, the glycolysis is stopped. And lactate is acid. So if you produce a lot of acid, it means a lot of lactate, you stop the glycolysis. What else? Feedback that's quite popular in the metabolism. Fatty acid synthesis. Fatty acid synthase and the acetyl coenzyme A carboxylase. These two enzymes are inhibited by palmitoyl coenzyme A. Palmitoyl coenzyme A, that's the end product, and it's able to inhibit the first reaction. And the same you have in the synthesis of purines and pyrimidines. Yes? They can inhibit themselves. The same you will learn in the synthesis of heme. Heme is a potent inhibitor of the enzyme, of the synthesis of heme. The second mechanism is the cross-regulation. It means that you have two pathways, and intermediate of one pathway influence the activity of some enzyme in the other pathway. So one example.
Is it purines or degradation?
That's purines, and how it works.
A of one.
Yes.
I don't think
This is more complicated example, but that's also true. In purine metabolism and purine and pyrimidine metabolism, we have both positive cross-regulation and also negative cross-regulation. The positive cross-regulation, if you synthesize purines, the purines activate the synthesis of pyrimidines. That's the positive cross-regulation. Yes? The negative cross-regulation is between two purines that AMP influence the production of GMP. Yes? The easy example is the malonyl coenzyme A. When we have fatty acids, synthesis of fatty acids and degradation of fatty acids, the intermediate of synthesis of fatty acids is the malonyl coenzyme A, inhibit beta oxidation, for it's able to block the carnitine transporter. Yes? So you can use two examples. The first, synthesis and degradation of fatty acids, or the metabolism of purines and pyrimidines. Two examples of cross-regulation. And the last is the feed-forward regulation, when an intermediate is able to activate something in the back. So do you know an example of the feed-forward regulation? And where? Fructose 2,6-bisphosphate, yes. This I do not think that it's the feed-forward regulation, but the fructose 6-phosphate is able to modulate the activity of phosphofructokinase. Yes. But that I also think that that's not the feedback or feed-forward inhibition. For fructose 2,6-bisphosphate is produced in the shunt of the glycolysis. When you have a lot of insulin, then you produce fructose 2,6-bisphosphate. That I do not think that it's feed-forward regulation.
What about acetyl-CoA and Kreb cycle?
Acetyl-CoA and Kreb cycle, and how?
I don't know that the substrate availability.
The acetyl coenzyme A, the definition is that the citrate synthase produces so many citrate. The definition is if you give substrate to citrate synthase, it produces the citrate. That I also do not think that is feed-forward regulation. I have only one example of this, yes. I found in the metabolism only one example. And it's in the Ugli pathway of purines and pyrimidines. If you remember, at the beginning of purine synthesis, you produce phosphoribosyl pyrophosphate. And the phosphoribosyl pyrophosphate is able to activate the further enzymes. Do you remember phosphoribosyl pyrophosphate or diphosphate? PRPP. Oh. I understand that the synthesis of purines and pyrimidines is not your favorite topic, yes? But that's the example of feed-forward regulation. The first reaction when you want to synthesize purines, you synthesize phosphoribosyl pyrophosphate. That's the molecule. And this molecule is able to activate the further enzymes in the synthesis. Yes? Is the potent activator of these enzymes. There was only this one example I was able to find in the metabolism of this. This is quite common, this also, and this is quite rare.
I think it's indirect feed-forward regulation and then fibrolysis
And where in maybe yes, but
Glucose makes the glucagon based from the glucagon binding protein in the nucleus. And
one more, the glucose makes the glucagon.
it releases from the nucleus the glucagon binding protein.
That I do not know, but maybe it's possible. Glucose makes glucagon.
No. There's glucagon binding protein in.
Glucagon binding protein.
Yes, that inhibits glucagon and it releases when.
The glucose.
Yeah, but
Then that would be the mechanism. But I do not know this mechanism, so thank you. We have two. But in the test will be nothing about feed-forward regulation. There will be only about the feedback inhibition. So good. Here you have also nice pictures, and that was everything before the break. So do you have any questions? And after the break, it's only your lecture for you will tell me all the regulations of all pathways. Now we have 15 pathways, so it means we have 2 minutes per one pathway. So the first pathway is respiratory chain and oxidative phosphorylation. So tell me the regulation. How is regulate respiratory chain and oxidative phosphorylation?
ATP-ADP ratio.
So ATP-ADP ratio. Here or something or not?
Substrate.
And substrate mean what? NADH and FAD. Yes? Proton gradient? Good. So there are two substrates of the respiratory chain. The first substrate are reduced cofactors, and the second substrate is ADP. So you need these two things, reduced cofactors and ADP, to have the respiratory chain. Then also you have to maintain the right proton gradient, not very high, but also not small. And one more thing you need to have, respiratory chain, oxygen. So we can say that the respiratory chain is controlled by the availability of oxygen, the availability of substrate, it means ADP, reduced cofactors, and then there are some factors that can regulate the proton gradient, for example, uncoupling proteins. So that was the regulation of respiratory chain. The second is the regulation of Kreb cycle. So how is regulate Kreb cycle? So we have 3 regulatory enzymes. Citrate or tell me it. Citrate synthase, then there's not the regulatory enzyme.
Oh, enzyme, sorry.
Isocitrate dehydrogenase, that's the key regulatory enzyme, and then alpha-ketoglutarate dehydrogenase. And how are these enzymes regulated? By some ligand or some substrate. So these regulatory enzymes are regulated by ATP and ADP ratio. If you have a lot of ATP, you inhibit these enzymes, yes? So ATP is inhibitor, ADP is actuator, and the same, it's true for the reduced cofactors. If you have no NAD and FAD, you stop the Kreb cycle. For these enzymes need these cofactors for their reactions, yes? So that was the regulation of Kreb cycle. 3 regulatory enzymes, ATP and NAD. Then we have pyruvate dehydrogenase reaction. So, pyruvate dehydrogenase reaction, it means what? What's the substrate and what's the product? Substrate is pyruvate, product is acetyl coenzyme A. But you know that to do this pyruvate dehydrogenase reaction, you need to have big enzyme, yes, that has many enzymes inside. So we are speaking about the complex of pyruvate dehydrogenase. For there are some subsequent reactions. There is no direct reaction between pyruvate and acetyl coenzyme A. So how is this complex regulated?
Again, ratio ATP.
Maybe some small influence. I'm not sure whether, but I don't think that that's the most important mechanism. So you need to have the cofactors and AD to do the reaction. For you need pyruvate and cofactor to produce acetyl coenzyme A plus reduced cofactor. That's true. But what's the regulation? The proper regulation. The first is that the product of this reaction, it means acetyl coenzyme A is inhibitor. So it's feedback inhibition. Yes, the product inhibits the reaction. Yes. So acetyl coenzyme A is inhibitor of this big complex. And then there is the second mechanism, that's the phosphorylation and dephosphorylation. And the question is pyruvate dehydrogenase complex active phosphorylated or dephosphorylated? Dephosphorylated. For when you have glycolysis, you want to continue. Glycolysis is activated by insulin, so the same for pyruvate dehydrogenase complex. So 2 mechanisms. And the question about this will be in the test, about the pyruvate dehydrogenase complex.
I'm sorry, could you please repeat, was it active phosphorylated or
dephosphorylated?
Okay.
Insulin, it means dephosphorylated. For if you look at it, that insulin activates everything to convert saccharides to lipids. And pyruvate dehydrogenase is in this line. You have glycolysis, pyruvate dehydrogenase, and synthesis of lipids. That's the insulin line in the metabolism. Yes, but remember the inhibition by the product, by acetyl coenzyme A. So it means that when you have a situation when you have a lot of acetyl coenzyme A, for example, due to beta oxidation, you stop this reaction. Yes? You do not destroy glucose and pyruvate. So we started here, respiratory chain, then we have Kreb cycle, now pyruvate dehydrogenase. So here we have glycolysis. So how is regulated glycolysis?
Ratio insulin glucagon.
So ratio insulin glucagon. Insulin activates glycolysis. It means that the dephosphorylation. Insulin dephosphorylation. What else?
3 key enzymes. 3 key enzymes.
3 key, what is it?
Glycolysis.
In glycolysis.
Glucokinase.
Glucokinase and yes. And how?
Pyruvate kinase.
So in the glycolysis you have 4 regulatory enzymes, or 3. The most important is phosphofructokinase. Yes? And insulin modulates its activity. What else modulates the activity of phosphofructokinase? Fructose 2,6-bisphosphate. What else?
Citrate.
Why and how?
Too much inhibits.
So, citrate is inhibitor. Question is why?
Because once it's accumulating in the mitochondria, then it goes to the cytoplasm and it's metabolized.
Yes, so when you have a lot of citrate in the cytoplasm, it means that you have a lot of citrate in the mitochondria. And if you have a lot of citrate in mitochondria, it means that there is the blockage in Kreb cycle. Yes, it means that isocitrate dehydrogenase is blocked. And glycolysis produces new substrates to Kreb cycle. So if you have inhibition of Kreb cycle, you don't want to produce new cofactor, new substrate for the Kreb cycle. Therefore, citrate is inhibitor of phosphofructokinase. What else? So we have insulin, fructose 2,6-bisphosphate, citrate, and the last, I've already mentioned it, pH. So low pH is inhibition. Low pH inhibition. That was about the phosphofructokinase. You know that the first reaction of glycolysis, that's the glucokinase or hexokinase, and that these two enzymes are regulated different. Yes? Hexokinase is inhibited by its product, by glucose 6-phosphate. So here we have it. Then the opposite process is gluconeogenesis. And you can remember that it's opposite, yes? Glucagon here is inhibitor, insulin is activator, so in gluconeogenesis is opposite, yes? So that you can, but there is one exception. The first enzyme of gluconeogenesis is called how? The first enzyme when you want to produce glucose from pyruvate.
Pyruvate carboxylase.
Pyruvate carboxylase, and this enzyme is modulated by what? By insulin, no.
No, biotin.
Biotin, that's the cofactor. That's the cofactor. For biotin is a cofactor of carboxylation.
Too much acetylcoenzyme A.
Yes. And inhibition or activation? Activation. So pyruvate carboxylase is activated by acetylcoenzyme A. So if you have pyruvate, you can produce acetylcoenzyme A, or you can produce oxaloacetate. Here we have pyruvate dehydrogenase complex. Here we have pyruvate carboxylase. And here we have inhibition. And here we have activation. Yes? So when you have pyruvate, you can degrade it, or you can produce from pyruvate oxaloacetate A. Oxaloacetate. This was very favorite question during the oral examination. Yes? The fate of the pyruvate and how the fate of pyruvate is influenced by the concentration of acetylcoenzyme A in the cell. And all other regulatory factors are opposite to the glycolysis. Only this is specific. Then we have metabolism of glycogen. So, metabolism of glycogen, that means degradation and synthesis of glycogen. The regulatory enzyme of synthesis is glycogen synthase, and regulatory enzyme of degradation, glycogen phosphorylase. How they are regulated? Insulin and glucagon ratio. If you have a lot of insulin, you have dephosphorylation and glycogen synthase is active and glycogen phosphorylase is inactive. Yes? If you have a stress condition, you have a lot of glucagon, adrenaline, you have phosphorylation in the cell, and therefore glycogen synthase is inactive and glycogen phosphorylase is active. Yes? That's the mechanism that it's quite simple and you know it. But also some intracellular cation is able to modulate the activity of these enzymes. Do you know what cation it is?
Calcium.
Calcium, and how and why? But magnesium also influences the level of calcium, so you can say that you have also
The release of glycogen starches in muscle.
Yes. So when you have the contraction in muscle, you need to have calcium and energy. So it's very nice if the calcium can produce energy for himself. Yes? That's the mechanism. If you rise the value or the level of calcium in intracellular fluid, what happens? You have some cascade. Calcium is second messenger. Calmodulin. So calcium binds to calmodulin. Then we have some calmodulin dependent protein kinases. Protein kinases, it means they do phosphorylation. So calcium, especially in the muscle, not in liver. Yes. So in muscle we are speaking about connection between contraction and glycogen degradation. And the connection is calcium, yes. Calcium means calmodulin, that means calmodulin-dependent protein kinase, phosphorylation of glycogen phosphorylase, and you degrade the glycogen. So that's the specific mechanism of regulation of degradation of glycogen in muscle, yes. Insulin and glucagon, they are more important for liver or for kidneys, yes. For these tissues, they can release glucose into the bloodstream. From muscle, you cannot release the glucose to bloodstream. So that was the metabolism of glycogen. And the last pathway of saccharide metabolism is pentose pathway. Pentose phosphate pathway, pentose cycle. So how is regulated pentose cycle?
Blocking on phospholipase.
Pentose cycle, that's the reaction. It has two phases. When you degrade glucose or not degrade, when you change glucose to some other saccharide. Yes? And in the first phase, you produce NADPH plus H plus. That this is this pathway. So do you know the regulatory enzyme of this pathway? Of the pentose cycle? What's the regulatory enzyme? First enzyme of the reaction, of the pathway.
PRPP?
PRPP, that's the Purines and Pyrimidines. But it was in the same lecture. You can say it, but normally in the books the pentose cycle starts with glucose 6-phosphate. Yes, but it's true that before that you have to produce glucose 6-phosphate. But when we start from glucose 6-phosphate, the first reaction is catalyzed by glucose 6-phosphate dehydrogenase. Yes, and in this reaction you produce the NADPH+H+. And the regulation is quite simple. You need to have NADP to do this reaction. So there is no specific regulatory mechanism, only the amount of NADP modulate the first phase. If you have NADP, you can produce NADPH+H+. If you do not have it, you cannot do the first reaction. The regulation of the second pathway, it means the interconversions between some saccharides, there is no specific regulation, yes? The only regulation there is that if you take some product from the second phase, you will produce new product. That is, for example, when you remove the ribose from that, you will produce new ribose. Or if you take fructose, you will produce new fructose. But the second phase has no specific regulation. The first phase, that's the amount of NADP. Now we have lipids. We'll start with the lipolysis. So how is regulated lipolysis? So in adipocytes we have triacylglycerols. How they are released from the adipocytes? By the hormone-sensitive lipase, and this lipase is controlled by insulin and glucagon. If you have high ratio insulin to glucagon, there is inhibition, yes? So hormone-sensitive lipase is inhibited by insulin. The second lipase that you know in the body is the lipoprotein lipase. And it has absolutely opposite role, yes? It's important for energy storage, to put new triacylglycerols inside. And therefore, the lipoprotein lipase is activated by insulin and inhibited by adrenaline and glucagon. So when we have lipolysis, the second step is to degrade the free fatty acids. So tell me the regulation of beta oxidation. Yes, malonyl coenzyme A, and how it works?
Inhibits carnitine transferase
1.
1.
So when you have free fatty acid in blood on albumin, then you put it into the cytoplasm and you have to transport it into the matrix of mitochondria. In the inner mitochondrial membrane, we have carnitine transporter. It consists of 3 enzymes, and the first enzyme is inhibited by malonyl coenzyme A. It means carnitine acyltransferase 1 is inhibited by malonyl coenzyme A. Malonyl coenzyme A is the intermediate of fatty acid synthesis. And then, of course, the beta oxidation is regulated by the amount of cofactors. For beta oxidation, we need NADP and FAD. Yes? So if you have no reoxidation in a respiratory chain, there is no beta oxidation. Then we have beta oxidation is connected to Kreb cycle, or the second possibility is the ketone bodies metabolism. So ketone bodies, they are regulated their production is regulated by the amount of acetyl coenzyme A in the mitochondria. So they are regulated on the step of lipolysis, it means hormone-sensitive lipase, and then on the step of fatty acid degradation, beta oxidation, it means carnitine acyltransferase 1. Yes, but there is no important regulatory mechanism of the ketone body synthesis. Yes, the regulation is on the step of lipolysis and beta oxidation. And regulation of fatty acid synthesis. So tell me the names of regulatory enzymes. So acetyl coenzyme A carboxylase and synthase of fatty acids. So we have two enzymes, the acetyl coenzyme A carboxylase and synthase of fatty acids. The key regulatory enzyme is acetyl coenzyme A carboxylase. And how we regulate this enzyme?
By citrulline accumulation. No, this is citric acid accumulation. Citric acid
Yes.
Activates.
Yes. So the first, insulin. Insulin is activator. Insulin is activator of the acetyl coenzyme A carboxylase. It means this enzyme wants to be dephosphorylated. The second mechanism is the accumulation of citrate. For the citrate has two roles in fatty acid synthesis regulation. The first mechanism is if you have a lot of citrated mitochondria, it is able to go into the cytoplasm, and there it's degraded to acetyl coenzyme A and then to oxaloacetate. Important is that the acetyl coenzyme A can go into the synthesis of fatty acids. So first, the citrate is able to give substrate to the fatty acid synthesis, and the second mechanism is that citrate activates the regulatory enzyme. Yes? And on the other hand, inhibits the glycolysis. And the last regulatory mechanism of this pathway of the acetyl coenzyme A carboxylase is the feedback inhibition by the product of this pathway, that's palmitoyl coenzyme A, or in general acyl coenzyme A. Yes? If you have synthesis of fatty acids, the next step is the synthesis of triacylglycerols. If you have accumulation of fatty acids, you don't want to produce new. So feedback inhibition by the palmitoyl coenzyme A. That's the last inhibition, the last mechanism. And then in general, you can say that if you eat a lot of saccharides and only lack of lipid, then these enzymes are induced. Yes, so saccharide-rich diet is inductor of these enzymes. And if you eat a lot of lipids, you have repression of these enzymes. Yes. Then we have cholesterol. So cholesterol that you remember from the last lecture. So how we can regulate cholesterol synthesis. Coenzyme A reductase. So the regulatory enzyme is called, of cholesterol synthesis, is the regulatory enzyme hydroxymethylglutarate coenzyme A reductase. And how we can regulate this enzyme? By insulin.
Ratio NADP.
Also you can say by cholesterol. What else? So here we have insulin, then NADP, then here we have cholesterol, tyrosine also, and if you are a doctor, statins. Yes. So insulin and thyroid hormones are activators. Or it's better to say in the when we are speaking about the tyrosine, there it's more the induction, so to change the transcription. Inhibitors are cholesterol and its derivatives and the statins. And two more pathways. The first is Urea Cycle. So how we can regulate the Urea Cycle? What? Carbamoyl phosphate synthetase 1. So the regulatory enzyme is carbamoyl phosphate synthetase 1. And how we regulate this enzyme? Yes, of course, that's the substrate. If you have no ATP, you cannot do the reaction. That's true. You know that there is some intermediate with stupid name, N-acetylglutamate. Is glutamate? Yes, glutamate. And this molecule is activator of the carbamoyl phosphate synthetase. But this you do not have to remember. Important is that if you have a lot of amino acids in the blood, also in the liver, that the level of amino acids, especially the level of arginine, they are able to activate this enzyme, yes? The direct mechanism is complicated through the intermediate, yes? But if you have protein-rich diet, it means you have a lot of amino acids, or if you have starvation, or you are in some catabolic state, for example, in sepsis or after burns or trauma, when the levels of amino acids when the level of amino acid is high, you have induction of enzymes of urea cycle. So that's the regulatory mechanism. So the more proteins, more amino acids in blood, the faster urea cyclase. So there's the regulatory mechanism. And then in urea cycle, we consume bicarbonate. And therefore, during acidosis, it means when we have very low pH, we stop the urea cycle. So the carbamoyl phosphate synthetase 1 is inhibited by low pH, by protons. The same as phosphofructokinase. And now these nice things, they are purines and pyrimidines. About the regulation of purines and pyrimidines, you should remember this first enzyme, phosphoribosyl pyrophosphate synthetase. And this enzyme is inhibited by feedback inhibition and inhibited by purine and pyrimidine nucleotides. Yes? This is the first enzyme in the synthesis of purines. And there is feedback inhibition between purines and this enzyme. Yes? There's the first and the second. The name of the regulatory enzyme you do not have to remember, but here only remember that the pathway, the synthesis of purines, and the synthesis of pyrimidines, both are activated by the phosphorybossyl pyrophosphate. That's the feed-forward regulation. Yes? In the first step, we produce something, and this is able to activate the other pathways. That's here. Regulatory enzymes of purine and pyrimidine synthesis are controlled by phosphorybossyl pyrophosphate. The same here. And they are inhibited by feedback inhibition by their products. Yes? GMP, AMP, the same here, UTP. And here you see the cross-regulation. It means that the synthesis of purines, it means ATP, activates the synthesis of pyrimidines. So that's the cross-activation. So do you have any questions to the second part, to the overview of all the regulations?
Good lecture.
So if you have nothing to do, then we should meet next week, I think.